Good afternoon. This is Dr. John Bennett broadcasting from sunny Miami. Today we have another in a series of stem cell science with Richard Mandel. He did part 1 last week. He's doing part 2 today. And we're also joined by our inestimable ally, Simon Downs from
That's a great word.
Hey, Simon.
Hi, thank you. It's great to be here. I feel very, very lucky indeed to be able to receive this lecture. I'm very much looking forward to it. Thank you.
Good. Welcome, Simon. Doesn't miss a session. Okay, Richard, welcome.
All right. Well, I think we'll just pick up a little bit before where we left off last time. We were talking about the holy grail with stem cells, and really that's a lot of what you were mentioning, Simon, in oncology now. It's the idea that with all the advances in immunology, that what we're going to be able to do is produce cells, tissue, organs that don't need immunosuppression, and they're almost basically personalized pluripotent stem cells. So I know that I'm trying to advance here.
Okay. You trying to change slides?
Yeah, it's,
okay.
Wait a second.
You might have to do it manually again.
What? Yeah.
Click on it. Yeah, there you go.
How's that?
Yep, there we go. Yep, it changed.
Okay. So we talked about aspirational stem cell therapy, and aspirational here is used as a euphemism, really. You know, as doctors, you think aspiration like aspirating a fluid-filled joint space or a needle, aspirating with a needle. This is more tongue-in-cheek, you know, meant to say aspiring to. So, you know, the stem cell tourism moniker, and we talked about Peyton Manning and the interesting use of at least the phrase stem cells in his treatment. And we talked about TV media and all the commercials, and I talked to you about a Neutrogena commercial that I'd seen one time where you know, talking about messenger RNA. It was mind-boggling to think that, you know, people are buying these skin creams really thinking that there was you know, a component of more than just cocoa butter in the thing. So we talked about the definition last time of stem cells, and remember that it was self-replication. It could replicate itself, or it was able to differentiate into many different cell types. But in a way, that's really very much shades of gray, because we talked about in vitro versus in vivo. And remember that in vitro is that's Latin for glass, and in vivo is, you know, within life itself, or like within the body. So the introduction today, the first really new slide is this: the fact that attracting attention and making promises results in money. So clinicians and scientists, you know, both have different perspectives. You know, we mentioned that unless you have a good understanding of the science behind the stem cells, that a lot of the attention that stem cell therapies attracted is more fanciful and kind of has gotten a lot more attention than really it was warranted. You know, to know the actual science of the stem cells, you really have to distinguish between mouse stem cells, human stem cells, in vitro and in vivo developments. And we also talked about last time that the embryonic stem cell itself, the totipotent embryonic stem cell, really doesn't exist in nature. I mean, we've never seen it. However, we have taken stem cells with a fair amount of pluripotency, not totipotency, and manipulated them in ways that in vitro, interesting things happen that suggest a lot of promise. But when we talk about in vivo, in life itself, we're talking about a much, much less abstract body of science. So let's look back here. Let's look the stem cell itself. Okay, so if we're talking about a quote-unquote stem cell in vivo, that's a lot different than something that's been taken and manipulated in vitro with things like trypsin. Trypsin's just something, an enzyme that you can lay down to have the stem cell kind of grow indefinitely over a layer of glass. What you could do by manipulating that cell in vitro in terms of radiation, in terms of exposure to toxicity of different things, is much, much different than what we really are capable of in vivo. So this is where the money comes in. By not having a good grasp of the basic science of in vivo cells, you can get conversations that don't make any sense, and come away thinking that maybe that's possible, when in fact it's still in the realm of years away. So this is an example where we're talking about totipotent cells. Human cells, the only totipotent human stem cell is that which results from the sperm and egg uniting, or oocyte uniting. And we get into shades of gray. John, we talked about this last time, where a lot of people have fallen into and it's become parlance now, the habit of talking about the embryo this, the embryo that. And the embryo is that product of the sperm and egg that makes up the fetus, and it's not what makes up the placenta. So we talked about, it's probably more appropriate, just technically, to call it the conceptus rather than the embryo, but nobody does that, right? So a totipotent cell is only that of the union of the oocyte and the sperm. Mm-hmm. So a pluripotent cell is basically anything else. So we don't really use this term totipotent anymore. It's pluripotent. So there's a tremendous variation of what falls in between totipotent and pluripotent. And then where we make the step into what we talked about before as renewal cells, cells that need to renew themselves. Now, not all cells in an adult human or anything else renew themselves. There are lots of things like the extracellular proteins in the lens of the eye, in the lens of our eyes, basically last a lifetime. That's why cataracts are a problem. I mean, if these were renewing cells, we wouldn't be getting cataracts. So you'll have blood cells that differentiate into red blood cells and white blood cells. The big difference here, when we think about it, is, remember, red blood cells have no nuclei, whereas white blood cells have tremendous nuclei and variability, and the ability to differentiate further into things like lymphocytes that we actually, have gotten very facile at manipulating. And then there's all these other things, which is what we talked about with the ability to differentiate in other cells. muscle, nerve, bone, quote, other tissues. So evidence. Okay, so a lot of times you'll listen to people and they'll talk about anecdotal evidence, and that's not evidence. It's just correlation or happenstance. When you can get people that'll say, "Oh, I used such and such, and my cold went away in 2 days." Well, you can treat a cold and it goes away in 2 days, or not treat a cold and it goes away in 2 days, or and so you can think that. Well, I guess the better way to say it is, the more variability there is in a disease, the harder it is to kind of predict its course, right? Common cold, whether or not you rub garlic on your head or took citric acid, probably doesn't make much difference. It's usually gone in 2 days, but cause and effect, that's real evidence. So when we start talking about applications for stem cells, we see all kinds of different things advertised. So, you have regenerative medicine issues. Now, there you have to worry about what the source is, and if there's a true effect, or if it's just a doctor feel-good remedy where there's an enthusiasm. You have a patient that's sick, and they want to believe something is happening, but it necessarily won't hold up to a appropriate clinical trial. Then you want to have controls, and of course the use of assays. A few years ago, a company named Geron, Geron, they were actually going to start a trial with stem cells to try to. It was a stage 1 trial, but it was still kind of amazing that it was going to happen. This is several years ago. It didn't. As far as I know, it didn't happen. I think it was slated to go off in 2009. Then it was delayed at least until 2010 or the middle of 2010. But I don't think it actually opened to enrollment of patients. As far as the embryos go, a few of the things I've looked at, mammalian embryos have a couple days of free living where the egg and sperm don't implant in the uterus. And, it's just a few days. And the point at which research or experimentation is done is when the primitive streak appears in embryogenesis. And that's where you get the 3 germ layers. Endoderm, mesoderm, and ectoderm. And that occurs at about 15 days. And pretty much uniformly throughout the world, it seems that the primitive streak is the cutoff point. And there has been some literature published about each religion, major religion's rationale. It was Christianity, Judaism, Muslim, and then a comment from Hinduism as well about their rationale. And like I said, I wasn't going to speak much about this at all. But if you want to look this up, there are references to why these religions think that this is the limit of experimentation. So remember that also when we're talking about these stem cells, you do have to distinguish whether it's a germline cell or a somatic cell. Obviously, a germline cell is a lot more important, as manipulating the germ line can result in perpetuation of the manipulations of the DNA, whereas in somatic cells, the differentiation may be much, much less apparent. So um, that's something else to keep in mind, whether you're talking about somatic stem cells or germline stem cells. So this was a very fanciful diagram of blast cell that actually is almost seems totipotent. And the take-home point here is just because you can draw it doesn't mean it can happen. It's still science, and it's still in many ways science fiction. But this does generate a lot of the ideas for what the particular targets are that take priority in terms of stem cell research. Many years ago, like 15 or 20 years ago, when when genetics and genomes were first being attempted to be quantitated, we thought that like cystic fibrosis would be a great disease that would be remediable with gene therapy, because it's a one-cell deletion, it's a one-cell problem, but it hasn't come as easily as we thought it could. So diabetes, because of the islet cells of Langerhans and the beta cells, that seems like a great target, a type 2 diabetic, an adult diabetic, where you can reimplant cells that will form insulin is great. That seems like something that's very, very doable at present. Whether it actually becomes that doable is another thing. We've heard many people talk about perhaps just being able to inject islet cells into the pancreas and have them function, but there's, of course, much more to it than that. That kind of harkens back to the surgeons in Mexico, the neurosurgeons who were implanting dopaminergic cells into the striatum years ago, and that was kind of flew in the face of a lot of basic science. So diabetes seems like a very good target now. Let's see, it's almost 100 years now. In 1919, there was a surgeon named Banning, and Banning, he was at the University of Toronto, and he had this medical student, very, very smart medical student named Best, and Best discovered insulin, and it was in 1919 that they did that. It was called Banning and Best. Banning, the senior researcher, put his name first, and that was okay. It was a major breakthrough. But Best went on to become an incredible physiologist, and the book Best and Taylor's textbook on physiology was the textbook of physiology for many, many decades. So Banning and Best. So it's almost 100 years since the discovery of insulin. And then the other big issue, another big disease, of course, is spinal cord injury. Now, as a neurosurgeon, I wouldn't have thought of spinal cord injury as a likely top candidate for the initial targets for stem cell therapy, because trauma to the spinal cord destroys a lot. You get a lot of glial scarring, and not only does it seem like a very difficult target to try to regrow neurons and oligodendrocytes, like myelin-producing cells, to spinal cord injury, but sure enough, Geron was on the brink of starting this trial on spinal cord injury several years ago. But I think that we're still in. There's a lot of basic science that needs to be figured out first. Parkinson's disease has seemed like a target for stem cells as well. The use of dopaminergic cells is to try to curb Parkinsonism is a good idea. The problem is that with Parkinsonism, it's a horrible disease, but it really doesn't shorten your lifespan, and the progression can be kind of slow. So it's going to be very hard for researchers to prove that the risk to the patient is quote unquote minimal, because it can take many decades to really get bad. And it's not going to be clear that stem cell therapy of Parkinsonism is going to initially improve the results that we get with deep brain stimulation with Parkinsonism. And then, of course, there's the problems with now the exploding field of neuroimmunology and knowing where to put those cells. Now, the other area that looks very promising for use of stem cells would be heart failure and myocardial infarctions. The idea that you could graft cardiomyocytes into areas that have infarcted seems like something reasonable that can be done. Osteoporosis and bone fractures seems like a likely target, but remember that bone and osteoporosis is very complex and hormonally. And just think of the work that Urbis did at UCLA in the 60s and 70s on bone morphogenic proteins. There's like 36 different proteins. That was in the 1970s. By now, there's got to be hundreds of precursors known. And then things like arthritis may be good targets because you could start with something like osteoarthritis and try to figure out some of the permutations that exist and the takeoff points into things like rheumatoid arthritis and other factors that contribute to joint disease. So we get now to this induced pluripotent stem cell, or IPS. This is technology that came out of Japan in 2006. This is this Shinya Yamanaka from Kyoto University. And what he did was he looked at cells that seemed to have pluripotency, and he looked at the oncogenes, as they were known then, cancer-causing suspects like OCT4, SOX2, KLF4, and c-MYC. Now, he terms them really transcription factors in the induction of pluripotent stem cells. With manipulation of these pluripotent cells, you get an idea of how these cells function in vivo, both in vivo and also in vitro. Now, this other term, somatic cell nuclear transfer, that's the initial technique that was used in cloning Dolly. Remember Dolly the sheep? This is the technique that was utilized, but it's been enhanced and advanced quite a bit since then. And now we're at a point where we're seeing some people that have mitochondrial diseases, and you're using the genetic material of a female with a mitochondrial disease with the male to create the union of what's usually the X and Y material, but they're using another female that doesn't have mitochondrial disease as really the bearer of the egg. And so in ways now that we are actually in vivo considering the implications of you know, 3 parents donating genetic material. So we talked about the targets, the potential therapies. Diabetes seems very reasonable now. Parkinsonism is mentioned frequently, although we're going to need to know a lot more about neuroimmunology. Heart disease, spinal repair, which I think is a great target, but to me it seems so complicated. I was really shocked that Geron was going to undertake this. And then retinal degeneration, you know, macular degeneration is a frequently mentioned target as well. So in terms of biology now and tissue-specific stem cells, you know, biology was traditionally like a non-quantitative science, but now there's going to be an explosion of quantitative information in biology, because as people start to study cell turnover in the body, embryogenesis, the renewal populations, the hemopoietic systems, and the GI tracts, I mean, the numbers are, you know, best thought of in scientific notation. I mean, for example, you know, the reason the stomach just doesn't digest itself is that the cell turnover of the stomach and the GI tract is so fast, it's probably faster than any kind of malignancy we know of, except maybe something like a Merkel cell tumor. But you know, the turnover's incredible. So, like I said before, drawing it doesn't make it so. Okay, so this is a good example of embryonic stem cells versus, quote, adult stem cells. There are not a lot of adult stem cells, and induced pluripotent stem cells can almost be used interchangeably with the phrase adult stem cells. So that's kind of an overview, and I don't think we really introduced a lot of new material. I think we just went and further defined what we kind of covered most briefly in talk number 1. I think that we can wrap stem cells up in 1, maybe 2 more talks that'll get into some more of the intricacies and the horizons that we're going to look at. But that's probably about it for today.
Okay. Very good, Richard. Thank you very much for a very clear presentation. Great slides. Really, really good slides. Just one quick comment before I turn it over to the students. I guess it's kind of the nature. It must be tough to judge when papers come out. I know that I have a hard time discovering, you know, figuring out if it's just promotion, fluff, because the nature of research, the researchers kind of have to exaggerate their I know they can't exaggerate evidence, but they kind of say breakthrough therapy, breakthrough, and they kind of blow it up to be, you know, such a big thing when it's hard to really say, is it really a breakthrough? You know what I'm saying?
I know exactly what you're saying. I really do. Because
to get money, they have to say something's happening.
Yeah.
Well, you have to remember that much of what goes on in science today is not science. It's just corporatocracy, where giant amounts of corporate money are just ravaging academic science, you know?
Mm-hmm.
And we are all, by definition, kind of clinicians who want to understand science. I mean, I wish I was a scientist, but I'm not. I just read all this stuff, right? Mm-hmm. So don't you know, there's a tremendous problem 2 or 3 years back where a Korean researcher claimed a lot of achievements with stem cells, and the 2 seminal papers he produced were later retracted. And there have been a lot of issues recently with the quality of research, but the things to look at are really reliable peer-reviewed journals like Science. Like I like the American Chemical Society. They produce great information all the time. It's not subject to commercial nonsense, especially, you know, when you get into medical, you know, papers in the medical literature. A lot more money's at play. But remember, for everybody here that pays taxes in America, you're entitled to an NCBI number, so you can apply to the you know, National Institute of Medicine and just get an NCBI number over the internet that gives you access to the current research. I mean, it's incredible. Having an NCBI number allows you access to some of the best journals in the world all the time. So PubMed, and just look up NCBI. I mean, I have an NCBI number that I use to look all this stuff up, but you want to try to review the big peer-reviewed journals. And I was reading about cancer immunotherapy and about all the progress we made with T cells that Simon was alluding to last week. And in oncology, you know, oncology's gone from being, the pillars of oncology have been surgery, chemotherapy, and radiation therapy for years. But now it's immunology is like the 4th pillar of oncology now, you know, with all the other medications, but the pharmaceuticals. But, you know, the way we advanced our ability to manipulate T cells is really, it kind of is science fiction compared to what we were able to do in the 80s. You know, it's taken 30 years, but the results are phenomenal. I mean, they've taken people with widely metastatic melanomas with a very short life expectancy, and some of these people have lost like 30, 40 degrees of their tumor burden and gone on to live for 2 years when, you know, they had a very limited lifespan prior to that. Mm-hmm. So, for instance, when I started looking at the T cell articles, I found the December 20th issue of Science from 2013, and in that they went through the breakthroughs of the year. And the breakthrough of the year at that time was cancer immunotherapy. This is a year and a half ago. And that's unusual in science to have such a clinical topic. But what do you think the runners-up were? CRISPR-Cas9 is what we just talked about a few weeks ago. So these are really well accepted papers that back this up. So I think, you know, you don't want to have to scour through thousands of journals. If you're an American, you go to Science. If you're European or British, you go to Nature, and then, you know, you look at the things that come from there. And you know, with an NCBI number, you don't even have to subscribe to either. You can read it all out of Nature or Science right on the NCBI.
You know,
or the American Chemical Society.
A lot of very reputable universities are doing stem cell. UCLA is doing a lot of neuro work. You're probably familiar with that. And Mayo is doing a lot of stem cell. So there is something to it.
Everybody's got doing research in stem cells. The idea is great. It's just, you know, you don't want to be fooled. It's not the academicians and the basic scientists that are advertising like Neutrogena facial rejuvenation. I mean but it, to the layperson, it must be very difficult to understand things, and that's why, as a clinician, you really have to keep an eye on what commercial entities are doing, because a lot of it's very, very specious.
Okay, Simon, do you have a question?
Oh, yes, please. Thank you very much, Dr. Mandels. As always, I'd like to know as much as I can about the subject. You talked a little bit about the pancreas. My sister is a researcher at UCSD, and she implants the beta cells into
The islets of Langerhans?
Yeah. I mean I think the beta cells.
Yeah.
Yeah, into insulin-making cells, and so that sparked some interest. But in the first lecture, you talked about how a lot of this is, it's mostly been done mostly in vitro, and in vivo, really, we don't really observe it, or we have proof of it. Just that we expect that this is what happened because we see what's happened in vitro. Which brings me to, I live in Japan, and recently the Japanese government has said we want to be the leaders in stem cell research, and we don't have the ethical issues. We don't need to talk about the ethical issues, but that the States may have, or Europe may have.
iTunes app needs your attention.
And what I was working at Baxter, teaching English and teaching the basic sciences English when the Mrs. Obokata scandal happened. If you remember, with Ricken, when with the STAP cells, and she published in either Science or Nature, and she generated pluripotent stem cells by subjecting them to stress or weak acid, which made them become pluripotent, and it was published in Science or Nature, and then they actually had to retract it. But the question is,
wasn't that that was from the Korean literature, wasn't it? It was a Korean.
It's Japanese. A Japanese Mrs. Obokata.
That was just last year, right?
Yes, she's from Riken.
I think it was recently.
And her superior committed suicide to take responsibility for that. But my question to you is, why was she blamed for it when a peer-reviewed article is, a lot of people take responsibility for checking peer-reviewed articles. Why is it that those checkers were not targeted also? Why is it she?
I don't know the answer to that particular case, but the one I read, there was something happened similarly in South Korea, and what they did was they talked about pluripotency being conferred on a series of fertilized cells, but it turned out that some of the data wasn't true. It was falsified because the cells really weren't fertilized, and so it could get pretty confusing.
But why would somebody falsify something? What could someone gain from falsifying that data if it couldn't be reproduced?
Academic, maybe an academic.
Yeah, to get funding.
Money, maybe funding. I mean, funding today is cut through, but yeah, and you don't want to blame the peer reviewers too hard, because they're not
following what she said. I see.
They're not the police. They're trying to advance the science, and I mean, corruption's everywhere.
Yeah.
Corruption is everywhere.
Yeah.
Being a politician or being in government is just legalized corruption. Any corruption outside of government is not legal. But there's always corruption. So you always have to be alert to the possibility of issues like this, and you always have to wonder where somebody's
this is a huge scandal.
Where somebody's motives lie. But I think in a place like Japan, I think places where your societies are more homogeneous, I think you can advance the science a little bit faster, because everybody. I'm not saying everything's correct. I'm just saying that a homogeneous society is not going to have the wide variations that a much more heterogeneous society is going to have. Like, I think that there was a company that came out of Iceland, maybe out of Reykjavik or something, but you know, the Icelandic people are very similar, and the genome's been contained, you know, reasonably well for many thousands of years. So I think that there were a bunch of genetic companies coming out of Reykjavik. So I would think Japan would be similar, but I am no expert on that. That's just my guess.
Riken is the biggest one here.
Yeah, shifting gears, Richard. I want to make sure I understand the cutoff point. 15 days. It's kind of, it depends on the religion, whether it's 10 days, 17 days. Some people call it a cutoff point earlier than later, and that's the point where a cell becomes differentiated.
Well, you know, it looked to me like the point at which you have a primitive streak, which is the embryo demonstrates mesoderm, ectoderm, and endoderm, the three germ layers, that everybody seemed to have a consensus that was where experimentation would not be tolerated beyond that. It seemed like like many of the major religions concurred with that for much different reasons.
Right.
But you know, I just saw that in some of the articles. But you know, the primitive streak in humans is at 15 days. In mice, it's like at 6 days. But you know, primitive streak for neurosurgeons is interesting because that's where we see the defects with myelomeningocele and spina bifida and the failure of closure of the spinal canal.
Right.
So, I mean, it's very early on.
Okay. Okay. Mohammed, you're there, Mohammed?
Yes. Thank you, Dr. Mendel, for bringing to us more details on like the stem cell. I have a question for you. I mean, I'm a true believer of stem cell therapy, but I always think when it comes to neurons, it's not like just putting a neuron, like they have to put a neuron with all the connections with different type of neurons, different structures. So I always think about how will they be able to accomplish this? Like how far they've been through this?
That's a great question because you have to remember I talked about the quantitative aspects of biology, which people would often think of as like a little bit softer science because you didn't have so much quantitative stuff going on. The fact is there's tremendous quantitative stuff going on, and you know, by the time you think of 2 billion neurons in your brain, you're talking about trillions of different connections. And what neuroscientists always say is you can't change one thing without changing everything. And that's the truth. That's why it seemed mind-boggling that spinal cord injury was going to be an initial disease treated because we really don't, you know, it's hard to understand just how well, like how oligodendrocytes are, how they function, you know, because they lay down all the myelin, and that's a complex process. They have to know what to myelinate, what not to myelinate, and when you think of neurons, you know, just when you think of like an action where you use an extensor muscle, you have to remember that while you use the extensor muscles, all the flexor muscles are being inhibited, and like glycine is an inhibitory neurotransmitter in the spinal cord, and so there's like 9 or 10 glycine synapses for every excitatory synapse. So everything you want to do, you know, you may have an excitatory pathway, but then you have 10 other inhibitory pathways to make it a smooth function. So yeah, the position of a neuron I mean, it's kind of mind-boggling. The mathematics of it must be incredible. I mean, I'm interested in that, but I am by no means any expert on this. I have no, you know, I look at it, but I don't pretend to understand, you know, any of that stuff.
Yeah, it's more than replication of the neurons, it's replication of the support tissue too, correct?
Oh yeah, it's not, you know, it's not just the neuron.
Yeah.
I mean, in this science issue from December 2013, the winner of the techniques in science that year was the neuroimmunology, the cancer immunology. The second thing was the CRISPR mechanism, which I told you about. And I'm sure that Dieteroff is going to get a Nobel Prize for this sometime. But the third thing was clarity, and it says clarity makes it perfectly clear. And what this is a system where the brain becomes, quote, as transparent as glass by removing the fatty light-screening lipid molecules that form cellular membranes and replaces them with a clear gel. So what you're left with, and I wish I could show you the picture here, is a brain that appears clear except for all the fiber tracts. And knowing those fiber tracts, you know, we used to talk about localization in the brain, and, you know, we may know where, you know, an auditory, you know, something is by virtue of where the nucleus is, but the fiber tracts that go all over the place is really the real science of how the brain's folded, wrapped, and everything else.
Mm-hmm.
It's very interesting.
Okay, Simon and Mohammed, any more comments,
It's
questions?
fascinating.
No, thank you.
Thank you, Mohammed. Simon?
Well, it's just the brain is the never-ending interest and fascinating. It seems like I can never get enough and never learn enough, and I forget things too, which I wish I wasn't. But I look forward to learning more about the brain, and thank you so much. I'm looking forward to the next one.
Very good. Go ahead.
We'll see where to go. I mean, let me look at, you know, what else should be in the syllabus for basic stem cell stuff, and I'll have that next week, and we'll see how far we get.
Yeah, maybe after that we can do neuroimmunology. You referred to it a couple of times.
Yeah, I think the neuroimmunology is really interesting, but of course, you know, to understand that, you have to know a little of the immunology, and I've been looking at that. So maybe we'll do immunology and then talk about neuroimmunology, because
well, Simon just studied immunology, so he can help us out.
Yeah.
Just finished my blog on that, so I'm very excited.
He just took that in med school.
I love to review it. I love to review. It's fantastic.
Very good. Okay, thank you, Richard, for coming out today and spending the time. Excellent. And thank you, Mohammed, and thank you, Simon. And we'll see you next week in our third installment of Stem Cell. Thank you very much.
All right, thank you.
Thank you, everyone.